Okay, so welcome to this next video in the playlist on experimental techniques. In this video, what we're going to talk about is antisense oligonucleotides, which are a way of controlling the expression of genes within cells. Specifically, they're a way of cutting off the expression of a protein within a cell of your choice. So antisense oligonucleotides. So these are a way of stopping the expression of a certain protein that you want to block the expression of within a cell. Okay, so the structure for this video is we're going to start off with the background knowledge, which is we're going to start off with a revision of the central dogma of biology. Okay, we'll then talk about what antisense oligonucleotides are and what they're going to do and how they're going to cut off the expression of a certain protein within a cell. And then we'll talk about the different examples of antisense oligonucleotides. Okay, so let's begin with the central dogma of biology. So we'll start with DNA. So in all the cells of your body, in the nuclei of these cells, you have 46 chromosomes, which are massive, great strands of DNA. Okay, and these chromosomes have a number of genes on them, basically, which are portions of this great strand, which is the chromosome, which are going to be used to make protein. So let's go over this. So basically, let's have a look at the structure of a DNA double strand. Okay, so we'll start here. So we'll draw out a little cartoon for the structure of it. Okay, so we'll go like this. So DNA is composed of nucleotides, known as deoxynucleotides. So I'm just going to draw a cartoon. I'm not going to draw the skeletal formula. Okay, now these nucleotides that are within DNA consist of a sugar, okay, which is deoxyribose. Okay, so let me colour in this deoxyribose sugar in blue here. So the sugar consists of a 5-membered ring here, where one of the members is oxygen. This tip of the ring up here, this member up here, is an oxygen, and all the other 4 members are carbons. And then you have the 5th carbon coming off up here, and that's also part of the deoxyribose sugar. So this is specifically 2-deoxyribose, and that means that there is a missing alcohol group coming off this 2nd carbon that you would have in ribose that has been replaced by just a hydrogen atom coming off there instead in deoxyribose. Okay, so then what you have off this 1st carbon of the sugar ring, you then have an organic base, and there are 4 organic bases that you have in DNA: adenine, thymine, guanine, and cytosine. So A, T, C, and G for short. Okay, so let's say this one is adenine here, just to give an example. Okay, and let's colour in adenine organic bases in pink here. Okay, and we'll also now colour in that final little circle that we had up there, which is the final component of a nucleotide. It's a phosphate group, which is attached to the alcohol group that comes off this 5th carbon of the sugar molecule by a phosphoester link. Okay, so this is a phosphate group here. Okay, right. So the entire structure, which is a ribose, or rather a deoxyribose sugar with a phosphate group and an organic base, which is one of these 4 here, attached to it, that's what is known as a nucleotide. Okay, so nucleotide. Now, what you do then to make a single strand of DNA, you polymerize a large number of nucleotides together. So let me show you how you go from one nucleotide to a single-stranded piece of DNA. Okay, so this 3rd carbon of the deoxyribose ring here, this has an alcohol group coming off it, and you can bind this via a phosphoester link to another nucleotide that's going to sit below it. Okay, so here then is the next nucleotide sitting below it, which is bound to the nucleotide above it, basically. So here we could stick on another organic base here, so we'll have cytosine here. Okay, so it's all the same again. You've got a phosphate group, a deoxyribose sugar, and then an organic base. So that's the single strand of DNA. And although I'm only going to draw 2 of these here, this will continue on basically. So you'd have more and more nucleotides following suit. Okay, and chromosomes can be, hundreds of millions of nucleotides all polymerized together, so they're massive great strands of DNA. Now, they do not exist as a single strand like this. Instead, they have bound to them a complementary strand, which means that it's going to have complementary organic bases in the analogous positions. So, let me show this. So, adenine always binds to thymine, okay, on the other strand. And they'll be held together by 2 hydrogen bonds between the adenine and the thymine. Okay, meanwhile, cytosine always binds with guanine. So this is a guanine here, and they're held together by 3 hydrogen bonds between the 2 organic bases. Okay, then this complementary strand, which has the complementary sequence of organic bases, runs what is known as anti-parallelly, or in anti-parallel fashion, to this DNA strand here. So what that means is that the orientation of the sugar-phosphate backbone, as it's called, which is the name for this portion without the organic bases, this portion to the left of my pen here is the sugar-phosphate backbone. The sugar-phosphate backbone is going to run in the opposite direction on this strand here. So it's going to be like this. Okay, so here is our deoxyribose sugar again. Here's the 5th carbon, and then we've got our phosphate group up here. Okay, now we'll put in the next one here. So here's our next deoxyribose sugar attached to this guanine here, and then here is the 5th carbon with the phosphate group attached to it up there. Okay, so this is what I mean by the strand runs anti-parallel to this strand here. That the sugar-phosphate backbone runs in the opposite direction, basically. So, anti-parallel. Okay, right. So, that's DNA. So, what are we going to do with the DNA? Well, what you will have in the DNA, so if we've got our double-stranded piece of DNA, which will downgrade somewhat now in the picture, we'll just denote it as these 2 strands here. It will go on and on and on for the entire length of the chromosome. Okay? Now, certain pieces of that, so let's say this portion here, will actually be used to make a protein, and this is what's known as a gene, basically. A portion that's going to actually be used to make something. Now, it's a little bit wrong to say that it's a portion that's going to be used to make a protein, because the definition of a gene actually is broader than that. It's a good starting point to define it as something that's going to be used to make a protein, but in reality there are some things that we would want to consider genes which aren't actually used to make protein, because occasionally the end product of a gene isn't actually a protein. Instead, you just want to make RNA from that gene, and the RNA itself is the end product. However, we'll exclude these little sort of minutiae. The general definition of a gene would be something that you're actually going to use to make a protein. But as I say, people and ah about what the definition of a gene is, and it is broader than that. But if you use that definition, you're not far off, okay? So, we'll define a gene to be something that we're going to use to make a protein. And then what you're going to do is you're going to produce a piece of mRNA from this gene. So here's our chromosome again. So what you're going to do is basically you're going to open up the DNA here, and you're going to synthesize a piece of mRNA, which is going to be complementary to one of these strands, okay? So, one of the strands of the DNA is going to be used as the template for the mRNA, basically, and one is not. So, the strand that you're actually going to use in transcription is known as the coding strand. So we'll say it's this one that I haven't bent, basically. This one that remained nicely straight. So let's say this one that I've now highlighted in red is going to be the coding strand for this gene. And I want to stress that there will be many different genes on this chromosome here. And if we say, for instance, take another gene up here, which I might call the green gene, okay? So let me highlight this in green. Okay, I'll get my green highlighter. So here's the green gene up here. The coding strand for the green gene might not be the same as for the red gene, so you might use this strand for the coding strand for the green gene. So it's not like one of the strands of the chromosome is the coding strand and one is the non-coding strand. It doesn't work like that. It's different genes along the chromosome will use differing strands, will alternate in the strand they use as their coding strand, okay? So we'll say that this is the coding strand for our red gene, okay? So what's going to happen is you are going to synthesize a little strand known as mRNA, which is complementary to that coding strand. Okay, now I want to discuss the structure of mRNA with you briefly. So, what is the difference between mRNA and DNA? Well, firstly, mRNA is always going to be, well, at least with regards to the central dogma, it should be as a single strand, basically. Okay, so you're not going to have it paired up with a complementary buddy, basically. It's going to be single-stranded, or at least it should be if it's going to actually be used to make protein. But the main difference with regards to the actual strand itself is that, firstly, the sugar that you use in mRNA, so let's show our sugar here. Well, let's actually draw a bit of mRNA out. So, if we imagine that this red strand, this coding strand of DNA, is analogous to this piece here, okay? So we'll be synthesizing a piece of mRNA that is complementary to this coding strand, so it will be running in the same way as this strand here, basically. So, it'll be running up and down. Okay, so, let's put these organic bases here. Now, the first difference is that you don't have thymine in RNA, okay? Instead, you put uracil where you would put thymine. So if we synthesize the piece of mRNA that will be complementary to this coding strand here, which we'll say is let's say this is a little piece of this red coding strand here. So let's now synthesize the piece of mRNA. Okay, so here we go. It's got a very similar structure, but the sugar that you're now going to put in here, the next big change between RNA and DNA is that the sugar is different. The sugar in RNA is ribose, not 2-deoxyribose. So let me colour this in a different colour. So we coloured deoxyribose in blue. Let's colour ribose in turquoise here. So, in turquoise, this is deoxyribose. Right, okay, and then if we continue on, it will then be bound up to another ribose sugar here, okay, and the main difference between ribose and deoxyribose is that on this second carbon of the sugar ring, you have an alcohol group in ribose, and in deoxyribose you don't have that. And then the organic base coming off this first carbon of this next one will still be a guanine. So in RNA, the organic bases that you use are adenine, uracil, guanine, and cytosine. Okay, and then it will still have its fifth carbon coming off here with a phosphate group. And the only reason that I've shrunk this organic base here is so that it will fit in. Okay, so that's the difference between RNA and DNA, but they're sort of minor differences. Okay, so you synthesize this piece of mRNA that's complementary to the coding strand of your gene, and what's now going to happen is this piece of mRNA is going to leave the nucleus after a few little modifications. It will leave the nucleus, and then it's going to be translated into protein. Okay, so here is a ribosome, let's say, and what's going to happen is that the RNA is going to go through the ribosome, so here is our RNA in blue, and what's going to happen is that the ribosome is going to look at the sequence of organic bases on the RNA strand. Okay, so it's going to look at them in triplets. So it will look at the first 3, and from those 3 it will decide what amino acid it should put in first, basically. Then it will look at the next 3 along here, and it will decide which amino acid it should put in second, and etc. It looks at triplets, okay? So it's what is known as the triplet code. It follows the triplet code. And these triplets of organic bases on the mRNA are what are known, is what is known as a codon. Okay, so a triplet of organic bases that's going to be looked at by the ribosome, and it will decide which amino acid that corresponds to. That's known as a codon. It codes for an amino acid. Okay, and every possible combination that you could have. Okay, so for instance, if we had an adenine in that first position, we could have then a C next, and then a G maybe. Okay. Every possible codon that you can come up with will have an amino acid that's associated with it, bar a few of them actually. Let me just explain this. So, firstly, let's think about how many different codons we could create. Well, we can put 4 different things in this position 1 here, okay? We could put 4 different things in position 2 here. Okay, and we could put 4 different things in position 3, and all of those possibilities would then be an individual codon. So there's 4 things that you could put in position 1. For each of those 4 things that you put in position 1, there are 4 things that you can put in position 2. So overall that gives you 4 times 4 possibilities, just looking at how many codons you could make if it just used a duplet code rather than a triplet code. And then finally, for each one of those, there's 4 different things that you can put in position 3, so then you need to times it by 4 again to get overall 64 possible codons. Okay, now there are 20 amino acids that are used in proteins, okay? So there is degeneracy, basically. What would be ideal is if there were 20 codons and then 20 amino acids, and then every single codon could code for an individual amino acid. Okay, it doesn't work that way. Instead, some of these codons will code for the same amino acid. So some amino acids will have multiple codons which correspond to them. And there are 3 codons which don't actually have any amino acid corresponding to them. And those are the stop codons. So when you get to them, then there's no amino acid that's going to be put in there, and instead the ribosome will stop the production of the protein, and that's when it finishes the translation. Okay, so that is our little brief discussion of the central dogma of biology. Okay, there is just another little concept that is going to be important for us, which is the concept of a sense strand and an antisense strand. So this piece of mRNA that we have produced here is what is known as the sense strand. Okay, now what does that mean? It means that the sequence of organic bases here is what can actually be read by a ribosome and turned into a protein. Okay? Now, if I instead made a piece of mRNA that was complementary to this non-coding strand, then that would make a totally different protein. It would make an utterly nonsense protein. Why would it make a nonsense protein? Well, it's going to have the sequence of organic bases, which is not the same as this mRNA, which is complementary to the coding strand, but instead is complementary to the sequence of organic bases on this mRNA strand. Okay, now when you take this other mRNA, okay, which we have made, which is complementary to this non-coding strand here. So I'll put this in green. Well, actually, no, I won't put it in green because I've already used green. I'll put it in orange. When you take this piece of mRNA and put it through the ribosome, it's going to have a completely different code, basically. It's going to be complementary, okay? So if this had ACG in position 1, which it wouldn't usually have. Usually you have AUG, which is the start codon. Instead, but if we stick with this one, instead on position 1 you'd now have the complementary ones. So complementary to A is U, complementary to C is G, and complementary to G is C. And the amino acid that you put in there may well be different. You might say, well, there is a chance that this is going to code for the same amino acid because of the degeneracy, the fact that some codons code for the same amino acid, and I would say you're quite right. But that's not going to happen for the entire code. You're not going to have that sort of luck. Somewhere it's going to be different. So the chances are that you'll make a completely different protein that will be utter nonsense, basically. So when you produce mRNA from a gene, you must use the coding strand rather than the non-coding strand. This piece of mRNA, which is complementary to the non-coding strand, or is complementary to the sense strand of mRNA, this is what would be called the antisense strand. So basically, the code, if you made a piece of mRNA which had the exact opposite, or the exact complementary sequence of organic bases on it to the sense strand, that is how you can make this antisense strand. And I will continuously in this video flip between putting a dash and then not putting a dash and just having them as the one word. It's trivial, basically. It doesn't matter whether you do put the dash there or not. So this is an antisense strand. Okay, so that's the concept of sense and anti-sense RNA strands. Now let's just think of it in terms of the actual DNA strands back here. So, if we look at this coding strand, that was complementary to the sense strand. Therefore, the sequence of organic bases on the coding strand of the gene was actually antisense, okay? Because if you'd used that sequence and plugged it through the ribosome, it would have coded for nonsense rather than something that made sense. Okay, whereas the sequence of organic bases on this non-coding strand, that's the same as the sequence of organic bases on the sense strand, because remember, they're both complementary to this coding strand. Okay, so the non-coding strand actually has the sense sequence of organic bases, basically. Okay, right. So, in the next video, we will now turn our attention to what antisense oligonucleotides are and what they're going to do.